Patel sir, former chairman NPPURC and ex-vice chancellor, Vikram University, Ujjain, Dr. Akhilesh Pandey sir, Dr. Vidhi Khurana from University of Delhi, respected faculty members, students and all other esteemed guests who have joined us in today's program of Indian Botanical Society Foundation Day Lecture, a prestigious academics occasion dedicated to celebrating the legacy, contribution and continued advancement of botanical science in India. I welcome all. Thank you, sir. Before we formally commence today's program, I invite you to take a brief look at the journey, vision, mission and objectives behind the establishment of Mansarovar Global University and Medical College through a short presentation. Presentation, please.
शिक्षा जगत की एक जानी मानी विभूति ब्रह्मलीन श्री कमलकांत तिवारी जी ने अथक परिश्रम और समाज के प्रति अटूट समर्पण के साथ सन 1995 में श्री साई ग्रामोत्थान समिति की स्थापना की। इस समिति की स्थापना का उद्देश्य था समाज कल्याण के साथ साथ वंचित और पिछड़े वर्ग तक उच्च गुणवत्ता की शिक्षा एवं चिकित्सा सुविधाओं को सुलभ कराना। इसी संकल्प के साथ सन 2001 में सीमित संसाधनों और मुट्ठीभर शिक्षकों के सहयोग से ग्रामीण क्षेत्र में मानसरोवर पब्लिक स्कूल की स्थापना की गई। यह तथ्य गर्व से कहा जा सकता है कि भोपाल मेट्रो के मास्टर प्लान में मानसरोवर स्कूल के नाम से एक स्टेशन भी प्रस्तावित है। श्री तिवारी जी की तपस्या और सतत प्रयासों से मानसरोवर की जड़ें निरंतर मजबूत होती चली गई और आज वे जड़ें एक वटवृक्ष के रूप में मानसरोवर समूह की विविध शैक्षणिक, चिकित्सकीय और सामाजिक शाखाओं के रूप में विस्तारित हो चुकी है। मुखिया मुख सब चाहिए, खान पान कहो एक, पाले पोसे सकल अंग, तुलसी सहित विवेक। आज यह कहना अतिशयोक्ति नहीं होगी कि परम पूज्य ब्रह्मलीन श्री कमलकांत तिवारी जी के प्रयासों का ही परिणाम है। मानसरोवर समूह एक प्रयासों का ही परिणाम है। मानसरोवर समूह एक परिवार की तरह हजारों परिवारों के पालन पोषण और भविष्य निर्माण का माध्यम बन चुका है। पेशे से इंजीनियर रहे श्री तिवारी जी ने 1980 के दशक में नेशनल टेक्निकल यूनिवर्सिटी ट्रोंडहाइम, नॉर्वे से उच्च शिक्षा प्राप्त की। वे चाहते तो विदेश में रहकर भौतिक संपन्नता अर्जित कर सकते थे, परंतु उन्होंने अपनी संस्कृति, संस्कार और मातृभूमि को सर्वोपरि रखा और देश लौटकर समाज सेवा को अपने जीवन का ध्येय बनाया। भारत लौटने के पश्चात वे मध्य प्रदेश विद्युत मंडल में अभियंता बने। उनका विश्वास था कि सपने वे नहीं होते जो सोते समय देखे जाए, बल्कि सपने वे होते हैं जो मनुष्य को सोने ही न दे। भारतीय संस्कृति के अनुरूप भावी पीढ़ी को शिक्षित करने का उनका स्वप्न ही मानसरोवर यात्रा का मूल आधार बना, जिसके लिए उन्होंने अपने जीवन की पूंजी का कण कण इस संस्था को समर्पित कर दिया। इसी क्रम में शिक्षा और चिकित्सा के क्षेत्र में निरंतर विस्तार होता गया। 2004 में मानसरोवर एजुकेशन कॉलेज, 2006 में मानसरोवर नर्सिंग कॉलेज, 2007 में मानसरोवर डेंटल कॉलेज एवं हॉस्पिटल, 2012 में श्री साई इंस्टीट्यूट ऑफ नर्सिंग साइंसेज।
इस बीच हमारे संस्थापक मेरे पिताजी ब्रह्मलीन आदरणीय कमलकांत जी तिवारी जी, 2010 में उनको कर्क रोग ओरल कैंसर हो गया और उस रोग से लड़ते लड़ते ही उन्होंने मानसरोवर के सफर को आगे बढ़ाया। मेरी माता जी ने उनका साथ दिया। 2010 के बाद भी मानसरोवर की लगातार प्रगति होती रही। हम लगातार छात्र छात्राओं की सेवा में, समाज की और राष्ट्र की सेवा में लगे रहे। 2015 में पिताजी और माता जी के द्वारा आयुर्वेद कॉलेज, मानसरोवर आयुर्वेदिक मेडिकल कॉलेज, 17 में श्री साई इंस्टीट्यूट ऑफ आयुर्वेदिक रिसर्च एंड मेडिसिन, 18 में मानसरोवर ग्लोबल विश्वविद्यालय, जिसमें कुछ जनरल संकाय थे जैसे जर्नलिज्म, मास कम्युनिकेशन, मैनेजमेंट, कॉमर्स, आर्ट्स एक्सेट्रा, वो चालू हुए। 2019 में हमने काउंसिल बेस्ड प्रोग्राम जैसे कि फार्मेसी, नर्सिंग और अदर एलाइड हेल्थ कोर्सेज को चालू किया। 2020 में मेरे पिताजी हमें छोड़ के चले गए और उसके बाद माता जी ने चेयरमैन और चांसलर के रूप में पदभार संभालते हुए मानसरोवर की इस यात्रा को आगे बढ़ाया। 21 में इस प्रांगण में आयुर्वेदिक कॉलेज की स्थापना हुई। 330 बेड के अस्पताल की स्थापना 2022 में हुई। 2023 में एनएमसी का एक नया एमएसआर आया जिसमें था कि आपको 650 बेड्स की आवश्यकता है। उस एमएसआर को फॉलो करते हुए इस अस्पताल को हमने 650 बेड का बना के 2024 में मानसरोवर मेडिकल कॉलेज के लिए एप्लीकेशन करी और आज हम अपने फर्स्ट बैच को इस कार्यक्रम में स्वागत कर रहे हैं।
आज मानसरोवर समूह के अंतर्गत विभिन्न स्नातक एवं स्नातकोत्तर पाठ्यक्रमों का सफल संचालन हो रहा है। इन समस्त उपलब्धियों श्री कमलकांत तिवारी जी की कल्पना, संरचना और तपस्या का साकार स्वरूप है। उनके द्वारा रोपी गई इस ज्ञान वाटिका की सुगंध आने वाली पीढ़ियों को निरंतर प्रेरित करती रहेगी। मानसरोवर की इस गौरवशाली परंपरा को आज श्री तिवारी जी की अर्धांगिनी आदरणीय श्रीमती मंजुला तिवारी जी एवं उनके सुपुत्र मानसरोवर समूह के मुख्य कार्यकारी निदेशक श्री गौरव तिवारी के नेतृत्व में नई ऊंचाइयों तक पहुंचाया जा रहा है। उनके कुशल मार्गदर्शन में मानसरोवर ग्लोबल विश्वविद्यालय को राष्ट्रीय एवं अंतर्राष्ट्रीय स्तर पर अनेक सम्मान प्राप्त हुए हैं और मोस्ट ट्रस्टेड यूनिवर्सिटी ऑफ मध्य प्रदेश जैसे गौरवशाली खिताब से नवाजा गया है। लगभग 125 एकड़ में विस्तारित इस विश्वविद्यालय में आयुर्वेद, कृषि, फार्मेसी, नर्सिंग, विधि, पैरामेडिकल साइंस सहित अनेक संकाय संचालित है। 1000 से अधिक विद्यार्थी यही शोध गतिविधियों में संलग्न हैं। शैक्षणिक उत्कृष्टता के साथ साथ सांस्कृतिक, खेल एवं रचनात्मक गतिविधियों जैसे पराक्रम म्यूजिक फेस्ट, युवा महोत्सव, उड़ान कॉन्क्लेव और एमसी रेडियो उड़ान के माध्यम से विद्यार्थियों के सर्वांगीण विकास पर विशेष बल दिया जाता है। देश विदेश की प्रतिष्ठित हस्तियों की उपस्थिति, अंतर्राष्ट्रीय संस्थानों के साथ हुई एमओयू और समाज के प्रति उत्तरदायित्व, ये सभी मानसरोवर समूह को एक विशिष्ट पहचान प्रदान करते हैं। धीरे धीरे सब कुछ हो गए, नारी सींचे सौ खड़ा, ऋतु आए फल बोले। आज मानसरोवर समूह केवल एक शैक्षणिक संस्था नहीं बल्कि एक विचार है, सेवा, संस्कार और समर्पण का विचार। ये उस यात्रा की कहानी है जो एक छोटे से संकल्प से आरंभ होकर लाखों जीवनों के भविष्य को संवारने वाली एक आंदोलन बन चुकी है। और यह यात्रा अभी थमी नहीं है, ये निरंतर आगे बढ़ रही है राष्ट्र निर्माण के संकल्प के साथ।
It's all about our Mansarovar. Now I respectfully request Chief Guest Professor Paramjit Khurana Ma'am, CDN Pro Chancellor Sir, Vice Chancellor Sir, Pro Vice Chancellor Sir, and Dr. Akhilesh Pandey Sir, please come on the stage to take their respective seats. Sir, please. We are deeply honored to have with us a distinguished academician and eminent scientist and innovative figures in the field of plant science, Professor Paramjit Khurana ma'am, to deliver the Foundation Day lecture, and we thank her for sparking her valuable time to be with us. Once again, I formally To formally commence today's program, once again I respectfully invite all the dignitaries to kindly proceed the lighting of the ceremonial lamp symbolizing knowledge, wisdom and enlightenment.
अच्युत शंकर प्रभृतिभिः देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। शुक्लां ब्रह्म विचारसार परमा आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदां जाड्यान्धकारापहा।
Thank you so much, sir. Now I request Honorable Engineer Gaurav Tiwari, sir, to extend a warm welcome to our esteemed guests by presenting a bouquet. Following the floral welcome, I humbly once again invite respected CD sir to welcome the chief guest, invited guest, and gathering with valuable and inspiring words. Sir.
सभी को नमस्कार। आज के इस कार्यक्रम में हमारे बीच पधारे आज के हमारी मुख्य अतिथि आदरणीय प्रोफेसर परमजीत खुराना मैम का मैं मानसरोवर समूह के इस प्रांगण में हम सभी की तरफ से, मानसरोवर परिवार की तरफ से हार्दिक स्वागत करता हूं। कुछ दिन केवल उस दिन के लिए लिमिटेड नहीं रहते, वो मेमोरीज बन जाते हैं। मुझे लगता है कि आज के दिन भी वैसा ही है। आई थिंक विल रिमेंबर दिस डे फॉर अ वेरी लॉन्ग टाइम, क्योंकि मैम की जो रेपुटेशन है इट प्रीसीड्स हर, और ये हमारे लिए ब्लेसिंग है कि आज वो हमारे बीच अपने सारे काम छोड़कर यहाँ उपस्थित हैं। एंड वी गोइंग टू गेट अ लॉट ऑफ विजडम फ्रॉम हर। साथ में आज हमारे बीच मंच पर उपस्थित आदरणीय प्रोफेसर अरुण पांडे सर, अखिलेश पांडे सर, प्रोफेसर AS यादव सर, कर्नल HR रोहिल सर, सामने की पंक्तियों में बैठे प्रोफेसर HK गोस्वामी सर, MK त्रिपाठी सर, राहुल श्रीवास्तव सर, NK प्रसाद सर, पुष्पेंद्र तिवारी जी, बाबुल ताम्रकार सर, सभी आयुर्वेद कॉलेजेस के प्रिंसिपल्स, डेंटल कॉलेज के डीन साहब, सभी फैकल्टी मेंबर्स फ्रॉम आयुर्वेदा एंड ऑल द अदर डिपार्टमेंट्स, डेंटल, फार्मेसी, फिजियोथेरेपी, एग्रीकल्चर। आज आप सभी, ऑफ कोर्स द डियर स्टूडेंट्स जिनके लिए हम सब हमेशा यहाँ पे मौजूद हैं, थैंक यू सो मच everyone for being here today with us. It means a lot. We look forward to the day and we look forward to gathering a lot of wisdom out of all of you. All these events, they make a lot of difference because this is when we get out of the textbook and, get to know about the broader sciences of the world. I thank NASI and co-organizers, Indian Botanical Society also for their support, and I think we're all ready for the enlightenment. And we're looking forward to hearing from our chief guest, Professor Paramjeet Khurana ma'am. Again, from the Mansarovar family and from all our hearts, we welcome you, ma'am. And thank you so much for taking out time today. Thank you.
Thank you so much, sir. To enlighten us about the significance, historical background, and objective of Indian Botanical Society Foundation Day, and also to briefly introduce our esteemed chief guest, Professor Paramjeet Khurana ma'am, with her distinguished academic career and notable contribution to botanical science, I respectfully invite Honorable Pro-Chancellor Professor Arun Pandey sir to kindly address the August gathering.
Chief guest of today's function, Professor Paramjeet Khurana, CED and Pro-Chancellor Engineer Gaurav Tiwari ji, Professor Akhilesh Pandey ji, Kulguru Dr. A.S. Yadav ji, Pro Vice Chancellor Colonel Ruhil ji. In the audience, I see Professor H.K. Goswami, our invited guest, Dr. M.K. Tripathi, Dr. Rahul Srivastava, Director Ayurveda, Dr. Babul Tamrakar, Senior Professor, Professor N.K. Prasad, our Registrar, Dr. Pushpan Tiwari ji, Principal of Ayurvedic Medical College, Dr. Anurag Singh Rajput, Dental College, Principal Gurdat Naik ji, Principal of Sri Sai Institute of Ayurvedic Research and Medicine, Dr. Manisha Rathi ji, distinguished faculty members, researchers, and dear students. I especially welcome Dr. Riddhi Khurana, faculty member in Hansraj College of University of Delhi. She is faculty member in the Department of Botany to Bhopal. About the IBS Foundation Day lecture, on behalf of the Honorable Chancellor of Mansarovar Global University, Mrs. Manjula Tiwari ji, CED and Pro-Chancellor Engineer Gaurav Tiwari ji, and on my own behalf, I extend my sincere gratitude to Professor Paramjit Khurana for graciously accepting our invitation to deliver the Indian Botanical Society Foundation Day lecture today. We are equally grateful to the Executive Council of the Indian Botanical Society for selecting Mansarovar Global University as the venue for this prestigious lecture. Allow me to briefly introduce the Indian Botanical Society. Established in 1920, the Society completed a remarkable century in 2020. To mark this milestone, it was decided that the Foundation Day lecture would be organized annually. The IBS Foundation Day commemorates the establishment of the Society and celebrates its enduring contributions to the advancement of botanical sciences in India. The occasion provides an important platform for scholarly reflection, dissemination of knowledge, and meaningful interaction among botanists, researchers, and students from across the country. As part of the celebrations, a special lecture is traditionally delivered by a distinguished scientist in the field of plant sciences. This lecture not only highlights contemporary developments in the discipline, but also reflects on the legacy and future directions of botanical research in India. Previously, Foundation Day lecture have been hosted at CCS University Meerut, University of Calicut, CFTRI Mysore, and CSIR National Botanical Research Institute Lucknow. It is a matter of great pride for us that the 5th Foundation Day lecture will be delivered by Professor Paramjit Khurana at Mansarovar Global University. It is now my proud privilege to introduce the chief guest of today's function, Professor Paramjit Khurana. Professor Khurana, Senior Professor in the Department of Plant Molecular Biology at the University of Delhi South Campus, is a name that commands immense respect in scientific circles. She completed her BSc, MSc, and PhD from the University of Delhi and subsequently joined the same institution as a faculty member, dedicating her career to excellence in teaching, research, and academic leadership. Over the course of her distinguished journey, she has twice served as the head of the Department of Plant Molecular Biology, 2004 to 2007, and 2016 to 2019, demonstrating exemplary vision and administrative acumen. Professor Khurana is a fellow of all 5 premier science academies in India and an elected fellow of the World Academy of Sciences, Italy. In recognition of her outstanding scientific contribution, she was honored with the prestigious J.C. Bose National Fellowship by the Department of Science and Technology, Government of India. With more than 4 decades of experience in teaching and research, Professor Khurana has made seminal contributions to the molecular understanding of plant growth and development. She is internationally recognized for her work in wheat biotechnology, sericulture biotechnology, and plant genomics. And her research has significantly advanced genetic transformation studies in legumes, trees, and cereals. She has authored over 200 research publications and successfully mentored 30 doctoral scholars, thereby nurturing the next generation of plant scientists. Her remarkable achievements have been acknowledged through numerous honors and awards, including the Professor Archana Sharma Memorial Award, Birbal Sahni Award, Dr. S. Rajam Outstanding Women Scientist Award, and the Women Leadership Award, among many others. She has delivered more than 300 invited lectures at national and international conferences, holds 3 patents, and has represented Indian science globally through plenary talks and academic engagements. Notably, she was also a member of a high-level Indo-US delegation to Washington. Professor Khurana has contributed extensively to science policy and administration. She served as a member of the Planning Commission's Working Group for Knowledge-Based Enrichment in the formation of 5-year plan. She was Foreign Secretary of National Academy of Sciences, India, and chaired the ACRB-PAC on plant sciences. I personally had the privilege of working with her as a member of this committee. She is currently INSA Senior Scientist from 2025 onwards and Vice President of National Academy of Sciences, India. Beyond her academic accomplishments, she has been a valued member and chairperson of several high-level committees constituted by the Government of India, reflecting her leadership, expertise, and unwavering commitment to advancing scientific research and national development. On behalf of Mansarovar Global University, it is my great honor to extend a warm and heartfelt welcome to Professor Paramjit Khurana. We are truly privileged to have her with us today. Thank you.
Thank you so much, sir. And now it gives me a great pleasure to invite Professor Paramjit Khurana ma'am to kindly deliver the foundation lecture and experience with us. Before we proceed, I request all the dignitaries to take their respective seats in front, please.
वो ले गया था वापस। वो ले गया था फिर वापस, शायद चेंज नहीं हो रहा था। मेरा ना एनिमेशन बहुत है, किधर करके करना पॉइंट। Anyways, good afternoon, everyone, and thank you so much, Dr. Pandey, for this wonderful opportunity to be present here at Mansarovar Global University. He's been insisting for quite a while that I come here, and I now feel that I should have come much earlier because it's such a nice, beautiful campus with lots of interdisciplinary subjects and other things as well. So I think it's indeed an honor, and thanks to the entire Tiwari family, I would say, to seed educational city. I would say this is a city complex itself, all the 3 campuses and all different disciplines of science, making it truly a holistic place to study, I think. So hats off to the visionaries and to the people who are now taking it forward as well. So indeed, it is a great opportunity to be here. And I also would like to thank and acknowledge the Botanical Society for choosing me with this particular honor of delivering the foundation day lecture. We've been botanists all. I think every naturalist is a botanist. Every person is a botanist whosoever, because you can't live without plants. So one way or the other, I think we are all related with the botanical species which are present on Earth. And it is indeed a great privilege to have been associated with Dr. Pandey when he was in Delhi University and see his commitment, his passion for studies of plants and sciences. During our various interactions at various forums, I've seen his commitment and his passion and his sincerity towards the discipline and subject. So he's a very respected figure, and very difficult to say no to him also. I've been delaying this. Okay. So earlier when we were students, we used to talk of DNA, a small fragment, a gene, sometimes RNA, if we are too advanced, maybe proteins, or the metabolites. But now the field has changed so rapidly that we now talk of the entire genetic complement of an organism. We don't talk of a fragment of DNA, we talk of the entire DNA of that organism, and that study is known as genomics. We don't talk of a single RNA strand, we talk of all the RNAs which are present in that particular time point, in that particular tissue, and that discipline is known as transcriptomics. Similarly, for proteins, we talk that as proteomics, the entire protein, not one metabolite, because nothing works in isolation. Everything is in a holistic way. And similarly, for metabolites also, the discipline is known as metabolites. So these are the various disciplines of omics, and there are many other intermediate ones also coming in. But what do they tell you? Why do you choose to study? The study of DNA or the genomics tells you what is the potential of that organism or that cell and what can potentially happen. Okay? That is the latent potential. In case of RNA, it tells you what is happening at precise that particular time point in that particular cell. And proteomics, which proteins are being present, are being synthesized in the cell at this time, what made them happen, what is their function, what are they doing. And for metabolites, there are many other things also which are present there and how they have come into existence. Next. So we say that plant sciences is witnessing an era of new change, new energies coming in, and we say plant sciences in the era of plant genomics in a big way. Next slide. So we were very fortunate to have been associated with this particular revolution at Delhi University, and India, in fact, it is an international effort, the sequencing of the rice genome. I'm sure you've all heard of the Human Genome Program. India, unfortunately, did not participate in the Human Genome Program. But when it came to plant genome, the secretary at that time of DBT, Department of Biotechnology, Government of India, she decided that India will participate, we will not miss the bus. She's entrusted us and another team at IRI to be part of this effort, and that was sequencing the map-based sequence of the rice genome to be there. Rice, because it's a major crop plant, and India decided it will be part of it. And this work was completed in 2005 and 6, and it made to the cover of both Nature and Science. Next slide. And because India participated, very rarely you'll find two magazines covering the same thing. And Nature and Science are always at loggerheads with each other. They exclude whatever is being published herein. But because the outcome was such a, considered as a landmark stepping stone in the area of plant sciences, that both of them covered it individually. Because of this expertise which then was gained by the Indians doing this work in India, in a public funded university like Delhi University, we were invited to participate in the International Tomato Genome Program also. In a while I'll tell you why tomato as well. And that work also was published in Nature. So after that next, we were also part of the wheat genome program. I'll be in a while why wheat was considered. In fact, when the rice genome program was written, it was said that the project proposal was initially for wheat, not for rice. But at that point of time, it was thought that the wheat genome would never ever be sequenced because it's a polyploid genome. It's a hexaploid as compared to rice, which is a diploid. And if you look at the genome size, it is huge. It is 16,000 megabases as compared to 420 megabases in case of rice. But then with advancements in technology going on during this course of time, it was possible to sequence even the wheat genome, and we were able to do that as well. In-house, and in-house I mean these three are the major international programs in which India participated in different levels. But in-house we also worked on the hot spring microbes also in Manikaran. That also made to the cover pages, and also the mulberry genome, that is the plant on which the silkworm feeds. Personally, my lab works extensively on mulberry. We have established mulberry also as a good plantation crop. So I'll take you briefly through this journey of ours which we have been able to do at Delhi University. Why I'm emphasizing again and again Delhi University is, we may be living under Indian conditions, working under suboptimal conditions, but when research comes, your research needs to be of an international level. And that is where we have to give in a more than 100%, I would say, if there is possible. So you can meet up to international standards as well. And we've been fortunate that the government has supported us in a big way, because without that, nothing is possible. Next slide. So this is the map-based, the first genome project which India participated. If you know, there are 12 chromosomes of rice, each listed in chromosome 1 to 3. And on top of that, you can see here different countries being donated. Like chromosome 9, there were 4 countries who sequenced it. And for chromosome 11, India participated in this particular program. Okay, chromosome 11. Now the interesting thing about this program is, this was a learning experience for us. No one had ever done plant genomics as such. No one had done any plant genome which was sequenced. So we all learnt on the project. But it was an international effort. All countries worked with the same standard. Okay, everyone had to sequence at the same timeline. The same timelines had to be followed. Every 6 months the progress was reviewed, and then the data was assembled. Now who would take up the assembly data? No one was willing to assemble so much data for the first time. But then India stood up to it. We said, okay, we will do it. And in a lab which was maybe 1/4 of this stage, we assembled this complete genome at Delhi University. And each molecule of DNA, it's one chromosome, is known as a pseudomolecule. And we assembled that. So it was a learning experience for us, and it was very satisfying for us to see that yes, we could work by international standards. Now, another interesting point of this genome project is that it is still today considered as the gold standard of plant genome. What does that mean? Gold standard means that the sequence which is available in the public database is so accurate that you will not find any single base pair change, at least in 1 in 110,000 bases. It is that accurate. And to get that level of accuracy, you have to sequence it multiple times, remove all the errors. Okay, so when you sequence one molecule, you've actually sequenced 200 times of it more to get to that uniform language and so forth. There is only one other plant genome which is of a gold standard, that is Arabidopsis, but that's a model organism. This is the only, till today, we have many genomes which have been sequenced today. They're all draft genomes, not complete genome. So this is a complete genome and with a gold standard as well. Next slide. This is a picture during one of the meetings where different country representatives come and they show the progress every 6 monthly when we meet. So here, next if you see, I particularly bring in this picture not only to show our Indian representative who's there. Next slide, if you, next. There is a lot of animation, yeah. So I wanted to bring in this to highlight this person here on the extreme left, the one in the maroon cardigan, Takuji Sasaki, the Japanese, who spearheaded this program. It was his vision that this rice genome needs to be sequenced, and he realized that Japan alone cannot do it, so he involved everyone internationally from different countries to do it. And it was his vision that yes, this needs to be there, and it was done. And additionally, this information is available in the public domain. That means anyone, you may have participated, you may not have participated, but you have access to this information. You can use the knowledge which has been generated as compared to the Chinese. When they publish the sequence, that is not available to you. You have to subscribe to that way. So this information is available in the public database, and it was basically, he is the visionary, the main force behind this particular effort. Next slide. So what happened after that? Okay, that was in 2006 when the rice genome was published. So using that information worldwide, different traits of wheat, the genes for different traits of wheat were understood. They were understood. Traits like heading date, which is very important for rice, seed width, seed shattering, seed length, GA insensitivity, racinosteroid insensitivity, the dwarf phenotype in rice, the silicon absorption capacity, the bacterial and fungal diseases, all genes of economic importance were uncovered, were discovered by different people worldwide accessing this information which is available in the public database. So this is the fallout of that, and this is not a complete picture. This is just a snapshot to show you what are the major traits, what is the output. जैसे आपने कहा ना कि धीरे धीरे चलते हैं आदमी, काम करते हैं, बाद में कुछ होता है. So the sequence was the first step. After that, so many genes of interest were understood, how they worked, and which gene is responsible for what trait. Next slide. So with this background information, we wanted to do something कि हमने अपने आप क्या किया. You know, your critics are your biggest critics, and you want to silence them. सबने कहा ये तो international effort था. You know, when people want to discredit you or not discredit you, do not want to give you credit. Okay, they'll say ये तो international effort था. So we said no, we'll do something in-house, and for the first time in 2006, a year after that, we sequenced the complete chloroplast genome of mulberry, okay, which was not an international effort. So this was the first complete genome to be sequenced from India, and it was done from my lab, and that is why the national flag there, because it was a matter of national pride for me. I said, nahi, hum kar sakte hai. You know, you want to silence your critics. Next slide. Next. So with the experience, and recently, before, say at the end of my retirement, we have also been able to, it took us 10 years to get funding to do the mulberry, Indian mulberry, the main genome as well. So we've been able to do that also, publish that also. And this is a comparative analysis of about 21 different accessions of mulberry, which have come from Japan, China, Thailand, and other places. Use them. It generated lots of SNPs, about 2.5 million of them, indels and others, which can be used now by others who want to do the breeding work. I'll tell you why. When we started work with mulberry, if you look at the NCBI database, there were only 7 ESTs reported, only 7. And from there, we have come to the complete genome of mulberry, both the chloroplast, this, and a lot of transcriptome data also, which I am not discussing today. But then this was very satisfying because we had traits which were species specific, which were tissue specific, because mulberry is a very interesting thing. The leaves are used for feeding the silkworm, the fruits are used for the pharmaceutical industry and others. I hear there are lots of pharmacy students also. They are hypoallergenic, hypocholesteremic, hypoglycemic, and hypocholesteremic in nature. One plant which has 3 major traits which are of use, and that is the mulberry fruit. And that is why I think it is very good, but the South Asian countries recognize it. India, it is not yet exploited that much. It is exploited only for feeding the silkworm. Next slide. So because of India's expertise, experience, I would say, now in the rice genome, we were invited to participate in the tomato genome as well. And we did the tomato genome also in another 5 years or so, and that also made to the cover. Because this is a fleshy fruit, that is a monocot grain. Next slide. But interestingly, if you look at the tomato fruit, there are about 7,000 varieties of tomatoes worldwide. Okay, but the genome is the same. So what is the difference? It is which gene is expressed in which species and how and when, under what conditions, what climatic condition. So it's a transcriptome which is important. So there is so much of diversity which is present in the genome, but is not necessarily expressed always. So that is where you unravel that what is the potential and what is being realized. Next slide. Interestingly, this tomato genome program soon transformed into the Solanaceae genome program, the entire family of Solanaceae in which tomato belongs. The reason being, in this family you have tomato, you have capsicum, you have sweet potato, you have potato, okay, two other major cash crops for other things. You have horticulturally important plants, you have medicinally important plants. Most of your vegetables belong to Solanaceae. But most importantly, you will find solanaceous members right from the dry deserts to the cold deserts. Okay? So when we say dry deserts to cold deserts, so as a plant scientist, what does it tell us? It tells us that genome of tomato had hidden conditions. You can extrapolate the tomato genome to capsicum, to medicago, to brinjal, and to other sugar plants also by comparative genomics. That's a different discipline slightly, but you can extrapolate it. It is like cousins. You know, family trait is one thing, then it runs in the family and you can extrapolate some bit of information. But major thing was that it had traits for adaptation to diverse environmental conditions. It can grow under dry desert conditions and it can grow under cool desert conditions as well. So it was very important to do the Solanaceae program also to understand the diversity and adaptation within different members in this family. Next slide. So a while ago I said we were able to do the wheat genome also. Initially the project was written for wheat, but they said it was never ever be possible. And they said कि आप please, DBT gave us instructions, wheat का तो पता नहीं कभी होगा कि नहीं होगा, आप rice पे program लिख दीजिए, project लिख लीजिए. And that is how we got into rice, honestly. And I was the most disappointed because I work extensively on wheat and mulberry. मैंने कहा यार हम तो wheat करने चले थे, आ गए हम rice पे. But that was blessing in disguise because we could not have handled wheat at that particular time. But after that, yes, we've been able to do that. We've been able to do for different tissues. We've been able to not only do it for the genome, but collate it with the transcriptome of wheat also for different traits and different organs as well. Next slide. Okay, so that's just what I'll talk of this. I'll come to why ultimately how do you use this information which is present here. The information which is generated is in tons, I would say, megabases and so forth. I'm sure you've all heard of the Green Revolution, then came the Gene Revolution in our country, and of course we are now heading towards the Evergreen Revolution. Next slide. So when we look at the food security next, the biggest challenge is what we feed is, number one is to feed people in 2050. Because of our population explosion, we must have enough food, but not having enough is important. Its availability, accessibility, and affordability is the most important. We have signed a trade agreement with US right now. Lots of things will be exchanged now further. They are the ones who really control which country will get what and so forth, but we have to make ways for that too. Next slide. I shouldn't have been talking of politics right now. So we all know that agriculture is sensitive to climate, okay? Any changes in the climate adversely affects the agriculture produce, whatever you may be producing. But let's also understand that because of agricultural practices, the climate is also changing. After all, agriculture is a monoculture. You cut forests, you cut lands to grow crops and others. And you spray lots of chemicals and fertilizers in the soil and you change the microflora forever. in there. So you are changing the climatic conditions as well, and there are lots of impact for that. Next slide. So the need of the hour is to have a climate smart agriculture. I want this to be the buzzword for next coming generations and for youngsters, that you must have a climate smart agriculture, because next, you should be able to efficiently utilize red, yellow is for soil, water, and your biodiversity which is available. You should be able to efficiently utilize these three of them. Next. So that, and then manage your ecosystems. Depending upon the ecosystems, you should be planting that plant there. It is not that you start planting pinus in the coastal regions and coconut in the hills. It will never be viable. So you have to integrate it with the ecosystems to have a sustainable livelihood. Next slide. So both of them go hand in hand, agricultural practices and whatever is available. And what is happening is we are outgrowing our earth. Why do we say we are outgrowing our earth? Next. Because if you see the food security challenge right now, the biggest challenge is that the water tables are going down and the temperatures are going up. Two environmental factors which adversely affect plant productivity inversely, both temperature and water, and we've tried to handle both of them, address both of them, I would say, by the biotechnological measures. Next slide. That's the title of a book, Outgrowing the Earth. Next slide. So, same thing, and this slide tells you about the politics, geopolitics of food security. Sometimes they say there is enough food for everyone, yet the plates are empty. That's a different economic aspect of food distribution and food security challenges which are there. Next slide. We'll not discuss that. Let's come to water once, and then we'll talk of temperature also quickly. What I've been able to do while addressing the water scarcity. Water is our life source, I would say, lifeblood. You can live without anything and everything except water. There is no replacement of water. Let's imagine. Even blood can be replaced. You can have artificial blood in it. Water, you cannot have it. And water is not only essential for us, but for the plant as well. No water, no agriculture, no food. No food, we are gone. Okay? And surprisingly, the earth is covered 97% by water, which is not usable for agriculture. It's only 3% fresh water which is available. Only 3%. And of this 3% also, you have water which is available and not available as well. Next slide. So water is important. Basically, it comes down to 1% of the fresh water which is available for agriculture. Next slide. So if we look at this as a satellite image picture, the real picture of the water content which was before we started this project, okay? And this was in the year 2000, I would say. Next slide. Next, if you zoom up, you see if you zoom here, the red color tells you where the water tables are going down more than a meter per year. This was in 2000. Now the situation is even worse. Okay, more than a meter per year, and this comes in this region known as the bread basket of our country, that is Rajasthan, Punjab and Haryana, which is your main growing conditions. Next slide. That is why we decided, and this is to show you that even crop plants have a water footprint. I'm sure you've all heard of carbon footprint, which is very popular these days. There's a similar term in agriculture known as water footprint. That means how many liters of gallons of water is required to produce 1 kg of that crop. If you see that, for corn, it's only 450 liters for a kilo of corn. But look at rice, 1700 liters of water is required to produce a kilo of rice. And that is why rice was grown in the coastal regions where water was prevalent. But the moment you shift rice cultivation from there to Haryana and Punjab, where water is scarce, the water tables are bound to go down. You may irrigate them artificially, but ultimately the water tables will get affected. Next slide. So we decided to work on water, and we looked at plants only, because there are many plants which will grow under low water also, without water also, and plants regulate it beautifully. Look at this. If the plant regulates it depending upon the atmospheric demands, kitni hawa hai, kis speed ki hawa hai, what is the temperature, what is the wind velocity, root, what is the soil, what type it is, what is the depth of the root, and then it regulates it. Kitna pani evaporate karna hai, kitna draw karna hai, and others. So the plants are very good in it. And there is one such protein. In fact, if you look at the seed, we say water nahi hoga to the plant dies. But in the seed, mature seed, the water content is about 10 to 20%. Yeah, yet the seed does not lose its viability. But you imagine a leaf coming down from 80% to 20%, it dies. It will not revive. So at the cellular level, if you see, there has to be a mechanism in the seed which protects the seed from death. Okay? And what is that mechanism? There are different types of proteins. One such protein is a LIA protein, which binds to the membranes during desiccation, so that as soon as water comes, they revive it and they form the original structure. Okay? That's a LIA protein. So you need to have your background basic information also. So we thought, let's try to engineer that particular gene in the leaf. If that seed protein is present in the seed and gives 20% protection, can give protection even under 20% water, what will happen if we produce that in the leaf? Will the leaf also be sustainable? Yes, next slide. And we had that engineered that in wheat for drought and salinity. I'm not showing the entire details. That's a decade's work. We had the HVA protein from Hodium vulgare. Because we were working with wheat, we were very sensitive about the fact we didn't want a non-crop plant gene to come in. So we took it from Hodium vulgare, which is also an edible gene, to wheat, which is edible, because biotechnologically you need to be very aware of your strategies, what you want to engineer, how you want to engineer. And we got drought and salinity tolerance in wheat. Okay, next slide. So we were approached by the sericulture people in Mysore. They said कि मैम आप हमारे mulberry को भी बना दो drought tolerant. So there was, and mulberry is a plantation crop, and means you grow it once, it will be there in the field for 10 years or so. They keep cutting the shoots and feeding it to the silkworm. It is the second fastest growing plant. एक दिन में आधा मीटर plant grow कर जाता है. So it grows so fast. And south में there is ample water. So they used to grow it very easily. Okay, no problem. But even in south, they're facing drought conditions, and good cultivable land is being blocked with mulberry cultivation. They want to use it for rice cultivation. So we said, okay, let's try. Let's put this gene in mulberry and see what. The problem was mulberry, no procedure, nothing. It's a tree species which is cultivated as a herb. Because you keep pruning it, keep using it, like it's a second after bamboo, it grows that fast. Next slide. Next. So to again cut a very long story short, we raised transgenics in mulberry, where if you see the first one is the non-transgenic. If you do not water it for a week, it wilts, the leaves are wilting. But in these three, they do not wilt if you do not water it also, because it grows very fast, so it needs lots of water for its growth also. And in the bottom line, if you look at the dry weight relative water content of the leaves, okay, focus on the blue bars. In the first one, that's the non-transgenic NT. The water content, if you do not water for 20 days, drops down to 35% or so. But in the transgenics, it is still at 75 to 80%, nearly double. Okay, so we said, very good. We have this good, and this work was done in 2008. So we said, कि हमारे पास कुछ है, and it is tolerant. Next slide. So we did all the analysis. We showed that it is drought and salinity tolerant. And at CSIRTI Mysore, that is the sericulture major headquarter in Mysore, they did not water it for 2 months also. हमने तो सिर्फ 20 days तक किया था. The plants would still survive. Next slide. So in this we did but the thing is, the leaf is the major thing. We can have the leaf right with the odd content, but the leaf quantity also should not reduce. Next slide. We'll show you. When we use this is the non-transgenic. When we use the actin as a promoter for driving this gene, the leaf area goes down little bit. Yes? So the farmer is not going to take it. Next slide. Next. But when we use a RDA promoter that is inducible to drought, it will get upregulated only when drought is there. We get different transgenics which are higher than the non-transgenics, the wild type, and one of them even double the size, the growth area of the non-transgenic also. So we did have salinity drought tolerance. We showed this also in 2011. Next year. Next. Similarly, once we had the right protocols in hand, they said, ma'am, humko ab disease resistance ke liye bhi de do. So when the farmer tells you to do something and you can do it, you want to do it also. So we did it for three different diseases of mulberry: leaf blight, anthracnose, and black spot, which are caused by different pathogens. And we showed that you can have tolerance Why transgenic approaches? Next slide. Similarly, we did it for beta-carotene hydroxylase. This is for high radiance, high temperature, and UV stress. So we have addressed salinity, drought, disease resistance, high temperature, UV stress, and high light intensity also. High light and UV, we got into this project because people in DRDO at Ladakh, they wanted something to grow in the dry desert area. And we said our mulberry is cold tolerant also, and it can withstand high radiance and UV tolerance too, so it will be able to grow there. So it was tailored for them. Next slide. So we had these traits here, we have tested them. Next, it's a perennial plant, so you can grow them. Next, we have tested them in our sericulture institutes and Delhi University. Next, transgenics in greenhouse, transferred them to GKVK in Bangalore. Next, where they are growing, now they become trees also, but they have to be pruned. Next. Okay, so that was the mulberry story. But unfortunately, now you'll ask me, why don't the farmers have it? For me, it was very easy. I could give a twig and give them for cutting, because you can vegetatively propagate them. The point is that policy, Government of India policy changed. We were very positive at that time because India was the only country, we had just sanctioned for the GM cotton at that time. And this would have been the second one. But then the policies changed in between and they said, no, we are not going to do that. So, okay, so we are stuck. What to do? So we started with the genomics aspect. The translation work did not work well, although they're already there with Mysore and Bangalore people. So little next chapter I will take you through the wheat genome also. Okay, I'm sorry if I'm boring you, but a little bit. This is the major crop plant, and I said wheat genome is important. It's a hexaploid, 3 parental genomes, 16,000 MB the size, and as compared to 3 PhD students failed. Someone said negative results are equally important, the positive ones here. So I'm telling you the importance of negative results also. Out of my 30 students, 3 students failed to isolate a gene in wheat because of the 80% repeated DNA in wheat. We were trying to do that in wheat, and every time we used to land in the repetitive DNA region and get nothing. So I had to change the projects in between also and get things. So we never failed. We failed, never got anything. And when we started this project, there were about 100 ESTs per megabase of the genome as compared to 3,000 which are present in rice and maize and others. And wheat is important to India. It's a major crop plant for us, and it is very sensitive to abiotic stress. And when we started this work, everyone laughed at us. He says, don't touch wheat, okay, because it's a huge one, and molecularly it's a nightmare. And we have practically seen that, yes, it was a nightmare. Of course, that student who did not able to get to the wheat genome is now a professor in IIT Delhi. Okay, that's another thing. But she did good work, did get the work we were looking. And we had the transgenics in wheat, all transgenics by Agrobacterium tumefaciens, the emmer wheat, which is a tetraploid. No, emmer is diploid, durum is tetraploid, and the bread wheat, which is a hexaploid. And at that time, ours was the only lab in the world, and CMET approached us to conduct an international workshop on transgenics in wheat, but DBT never gave us the funds. Okay, so because of international program workshops are much more intensive, maybe they were limited by their own funds and so forth. Next slide. At that time, we also had a transgenic in wheat, which till today stands to be the only transgenic in wheat for cereal cyst nematode. Okay, cyst nematode, there was a special workshop only on nematode biology in Turkey, and I was the only person invited from outside the CMET group of institutions, because we were the only lab which had this transgenics, and we showed that with transgenics with nematode resistance, see, you could get tolerance even when the nematodes are there. And nematodes, nearly 70% of the Indian fields are infested with nematodes, and they cause a heavy damage here. We said we showed them a procedure, but then it's a crop plant and our biosafety policies changed. Next slide. So we could never take it to, when you have a transgenic, when you want to give it to the field, they say, hame pure lines though. The geneticist will say pure lines. Now, as a molecular biologist, having a pure line is next to impossible. There is nothing known as pure line, okay? But the closest we could do is we could raise haploids, diplodize them, make a doubled haploid, where the entire genetic complement would be identical. We now have these transgenics in double haploid. Raising haploids is only 2% efficient in wheat. Transgenic is also 2%. 2% of 2%, 0.02%. And then we had the transgenics out of that, so 0.0002% efficiency. We still got some transgenics in this. Diplodized them, next slide. And had the next slide, all double haploids we call them, and tested them for salinity. See, 400 millimolar of salt. The non-transgenics will not even germinate, the transgenics will all germinate for salinity and drought both. Next slide. Not only in vegetative stages, even with crop productivity. I know it's a little complicated slide, but if you look at the lower bars, this shows you percentage reduction in seed weight, seed size, and total number of spikes per wheat. So you'll find that the percentage reduction has reduced by half. It's about 25% as compared to 60% or so in the others. Next slide. We have double haploids next. We thought that now we have the pure lines. We talked to the breeders in there. We wrote to many of the breeding institutions of working on wheat. If you could take them as, no one would touch transgenic wheat. And unofficially we were told, please do not advertise your work because our export will get affected. We export a lot of wheat, and the moment we say we have transgenic wheat, that might get affected. But no one was taken to. But then we thought that we have a wonderful germplasm in a hand where one trait was different, and we wanted to study what one gene could do in that entire complement. And it's a pure breeding line. It's a pure line. We have it now in the 10th or 11th generation, and we showed that besides drought and salinity, next, it is also, we checked it, click again. There's a lot of animation in my slides. I wish I had the tick here. Next slide, next Guru. It's also tolerant for heat stress tolerance, and heat stress is very dear to me and to all of us. But we could not do many things, so I say wishful dreaming. Next slide. So, next. So we changed our focus of work, direction of work, looking at temperature, warming predictions. Next slide. Everyone knows the world is warming up. Next. When we started this work, that was in 2007 or so. In 2006, the temperatures has gone up in this region, and temperatures rose by more than 5 degrees in this region. Now, why was this 5 degrees increase important? Next. If you see, this increase in temperature occurred in the period of February to March, just this time of the year. This is the time when the spikelet from the vegetative phase in the wheat. That means the plant shifts from the vegetative to the reproductive phase. And every degree increase in there, next, it brings down productivity by 4%. And for temperate crop like wheat, next, comes to 6%. So imagine in 2006, if the temperature rose by 7 degrees, how much wheat did we lose only because of heat stress? 42%, nearly half. Only heat stress. We're not talking of pathogens, we're not talking of other diseases. And I don't know, at least the people sitting in the front row, the seniors would remember, last time when India imported wheat was in 2007, after this break. And that is when DBT said, jaldi jaldi heat tolerance pe projects do, heat tolerance pe projects do. Because in entire India, the wheat germplasm which was there was bred for fungi, for Puccinia, उसको क्या बोलते हैं? हाँ, नहीं, disease का नाम क्या बोलते हैं उसका? Rust, for rust resistance, but not for abiotic stress, not for heat tolerance. There was no one was known. Nothing was known. कि किसी को पता ही नहीं, man, सारे rust वाले, yellow rust पूछ लो, orange rust पूछ लो, black rust पूछ लो, all the varieties were bred for them. So even the germplasm did not exist. No one knew what was there. So we decided, okay, let's take a dive. Next. And we realized that, next, if you see, we looked at the growth stages of wheat. Next. So we found that there are 2 stages which are critical. Heading stage, when the spikelet emerges, yes, in the northern region. But in the southern region at that time, the wheat is still in the tillering stage. So you have to integrate your growth and development with your plant sciences and genomics approaches. Everything is all integrated plant sciences, I would say. So there in south, it is tillering which is sensitive to heat stress. Now, if you have something for heat stress, what is working at the heading stage will not work there in the strains which are grown in south. and what is working there will not grow here. So you must have multiple strategies for different developmental phases of wheat to withstand heat stress, right? Next, that's what we did. And ऊपर से the wheat breeders told us कि मैम fertility भी compromise होती है. In fact, 60% reduction in anther viability. So हमने कहा अच्छा इसको भी address करेंगे साथ में. We'll take this also next. So we took up all these stages, and at that time internationally I told you wheat genomics was not available, nothing was there. What was available internationally in the international to TSC mapping work was EST production, objective 1 and 2. And they thought कि आप ESTs बनाओ उससे फिर it will feed into mapping में, functional genomics में या structural genomics में and other things. And we said okay, we will also be participate to it, and we did that on our own. Next slide. So again, I'm not telling you good work of another 10 years or so. We took up those vegetative stage, the heading stages, the flowering stages, anther and the others, and we did the transcriptome analysis. Also, we published lots of papers there, but what did we see? Next, if you keep on, the करते जाओ थोड़ा जब तक पूरा light up करेगा. So the entire cell gets reorganized under heat stress. The transcription factors getting up, there were post-translational controls, there were translational checkpoints, post-translational checkpoints and everything which was there. So we didn't know where to start. And when I presented this work in Russia, at St. Petersburg, an Australian scientist, he said, young lady, at that time I was younger, he said, now you have a big task ahead of you. I said, what? He says, now you have to tell me the function of each and every gene. I said, मर गए हम तो कैसे करेंगे मतलब we had thousands क्या millions of genes which are, how do we say which is important? He says, no, you have to do that, otherwise what is the point? I said, okay, we'll take that challenge as up also, do it one by one. Next. So every gene which we thought was important, we tried to functionally show its value, we show where it is important and what process it is important, okay, and we had good heat shock proteins. We've characterized each one of them. किसी में एक पेपर है, किसी में दो हैं, किसी में तीन है, एक ही जीन के ऊपर. Okay, we have lots of them. We have patents there. We've done the functional genomics, जिसको कहते हैं, for transcription factors, HSF2D, which is the master regulator, zinc fingers 2, Matchbox2, BZIP proteins for intracellular signaling, a brassinon steroid, a B2 protein, a VAP protein, lipid transfer proteins, STIs also, different osmolytes and so forth. Not trying to impress upon you, but trying to say that we have about 24 genes which have been very well characterized, and each one contributes to heat stress tolerance. So what do? So we were lost. We don't know where to go. We had lots of data, lots of information, lots of publications too. But when we went to DWR, that is in Karnal next, so I was faced with a very curious problem. And that is when you interact with the farmers, he's not impressed with your papers, your publication. आपने जो काम किया मैम आपकी लैब में किया ना, वो कह रहा है मुझे तो ये बताइए कि मैं अपने खेत में कौन सी लगाऊं. And can your information be used to? So you have to come down from your ivory towers back to the field and tell him how you can utilize this information there also. So there were two major questions before us which the farmers raised. Next. Number one, कि हमें ये बताइए which variety is sensitive and which is tolerant to heat stress. हम उनको grow करें फिर. Okay, we didn't have anything in the germ class. Second, अगर heat stress आ जाता है मेरी field में, उसके बाद मैं क्या करूँ? You know, when it is at the fruiting stage, seed stage, replant, उसको आप नहीं कह सकते कि ये variety नहीं grow करो, ये करनी थी तुम्हें. He says, what do I do? So we said, okay, these are two very basic questions and we tried to address them in very simplistic way. Next slide. Using the information we had generated, we created a heat susceptibility index. We screened about 13 ruling varieties of wheat, used different criteria at the vegetative stage, seedling, mature. FV by FM is the photosynthetic efficiency, membrane stability index, chlorophyll, thermal induction response, carbon, proline and others, and we made a heat susceptibility index and we said these varieties are relatively tolerant and these, the green ones are relatively sensitive to heat stress. हम tolerant नहीं कह सकते, resistant नहीं कह सकते. So you can say relatively tolerant and they have proved till today that yes, they are doing better, C03 and others, ऊपर की 4 and others. Next slide. Next, addressing the second question, these are the two tolerant ones जो हम कह रहे हैं कि tolerant है और ये हम कह रहे हैं कि sensitive है ज़्यादा. Next, This is how the seed looks like. जब heat stress मिलता है ना, this is how the grain looks like. So I'm not showing any data here, I'm just showing you the picture. If you look at the second, एक वापस जाइए. If you see the grain size 2329 में, will anyone accept that as a grain, wheat ka? It is more for fodder then. You know, after heat stress, the grain is transferred to fodder industry, not for human consumption. और tolerant में फिर भी थोड़ा है. So we were working on a different project which was affecting seed size and seed vigor. So we said, try करें इसको field में spray करके. It's a very expensive compound, but we said after the crop has seen the heat stress, what if we tell the farmer to spray this? Agar this molecule is increasing seed size or not, will it do that? Next. Now you see, from second to third, the seed size has increased. It has increased. In fact, it is nearly same to the control. Not so much in these, but फिर भी 60-70% तो increase हुआ ना उसमें. But unfortunately, this chemical we cannot advise them to give it. It's a very expensive chemical, good for lab use, but not for field use. Okay, so we're looking for alternatives for that. Next. And we then also started looking at other heat adaptable traits which provide photoprotection, which affect partitioning of the photosynthate and light interception. And we focused on this radiation use efficiency. Next. Because in radiation use efficiency, I'll tell you, we had some very interesting data. We had also shown that their thermostability and other things are present here. Next. So, and we combined this with, always I've been saying wheat has 3 genomes, right? So imagine this population, all diversity because of 2 genomes. No, everyone sitting in this auditorium is a genotype, right? Two parents. So much diversity in this planet. Imagine जब 3 parent होंगे कितनी diversity होगी. That is the amount of diversity, genotypic diversity which you get in wheat. But if you look at the parental genomes, the A genome, the B genome, the tetraploids, and this, by comparative genomics, you can study what is what. I'll tell you why we are important, why we came to the parental genomes of wheat. Next. Because one of my trips to PAU, Punjab Agricultural University, they had the germplasm of Aegilops. This is the donor of the A genome. Both Aegilops toshii and Aegilops speltoides. And in the month of June, these plants are growing. June in Punjab is 45 degrees plus in the field. And these plants are growing very well. So we brought both of them to Delhi, gave them heat stress. So we saw speltoides is susceptible, पीली हो जाती है, टोशाई नहीं होता, अभी भी green रहता है, दिल्ली में भी 47 डिग्री टेम्परेचर पे. So there has to be something. अगर ये रह सकता है, तो A genome is gone into hexaploid. We bring a comparative genomics into picture and see what has to be done. Next slide. So what we see is, if you look at, physiologists use this term very often, the chloroplast partitioning, PS2 activity, photosystem 1 and photosystem 2, all photosynthate is, electron is transmitted in via photosystem 2. So next, if you show, in case of toshii, if you see PS2 activity is 76% under normal conditions, under heat stress still 73%. But in speltoides, that 76 goes down to 26%. So in the physiology of the plant, it is the PS2 activity which is getting affected most, and that is what makes the plant susceptible to heat stress. Okay, next slide. We check that for a wheat varieties also, जो हम कह रहे हैं कि tolerant है और हम कह रहे हैं sensitive है, उसमें भी यही होता है क्या? Next. You will see same thing, the tolerant one, PS2 activity is still in the 70s, while in the sensitive it has dropped down to 25% or so. So we knew we were on track. Next. So why I'm saying this is, we had done the transcriptome of another important issue in the spike, which is known as the ON. एक लंबा सा seed के ऊपर से निकला होता है tissue, and that is also photosynthetically efficient. About 20% carbon comes up from there. So we looked at some of the genes, 2 homologs. Next, one is getting upregulated, one is getting downregulated. This is RBC rubisco activase B or A. Okay, next. We validated them. Next slide. PBW is 3,4 is the sensitive one. This is the control. RCAA दोनों में ही low रहता है, but in the tolerant one, RCAB isoform, that is getting upregulated. Next. Okay, both are isoforms. But one isoform is getting upregulated with heat stress, in the other it is not. And this is in the one which is tolerating it. That means RCA is the major player in PS2 activity, and we know it is a essential component. Next slide. So we looked at this gene family. Now in wheat, every gene has a 3 genotypes. It has about 6 alleles. Every gene has it. So you should know which allele you want to pick up. अगर आपने silent allele pick करके transform किया तो बेकार. You need the active allele. So we look at the profile in the leaf, the spike and the grain. और जो लाल वाला है ना, that is the one which is upregulated. So we choose this particular, these 2 alleles, which can be now engineered, which are important for heat stress tolerance. And we said, okay, if this is important, let's check it in another crop plant, that is rice. Next. So we now transform rice with the wheat gene and show express hota hai transgenics ka, and look at the photosynthetic efficiency under heat stress. The light green bars, these are all the L8, 9, 10 and 12 are the transgenics, and they are upregulated. That means they are not getting effect. They are tolerant to heat stress. Next. So picking the rice allele is important, and even at the grain filling stage, at crop productivity, the transgenics do better. Next. The number of sterile florets goes down. Remember, a while ago I started with heat stress affects sterility. Next, fertility rate increases. Okay, so we have the proof of principle that yes, this is possible, and with transgenic technology we can do this, and this work has also been published. Next. So, besides the heat stress, now we know more of the heat stress biology. We know that there are epigenetic controls also, which are important. We have 2 publications on that also, but I'm not going to talk more. Next. I think next. Basically, we now know a lot of the heat stress. There are, one thing is acquisition of tolerance, and the other is long-term adaptation. Okay, we have been focusing on acquisition of tolerance, कि कैसे tolerance integrate करेगा, and what are the key players. Many of the colored ones we have already worked on in detail and shown their mode of action and created something. A little bit onto the histone modifications and also chromatin remodeling for long-term heat stress tolerance, but still a long way to go. Next. Under non-transgenic approaches, you can see the later part I'm using all non-transgenic approaches. We've been also trying to do rhizosphere engineering, because certain bacteria are known to give protection against drought, heat, salinity and others. Yeah, this is my last slide. And couple that up because they provide not only nutrients but other stress factors also. Next slide. This work is not yet published, but we have some interesting data in the pipeline, maybe another year. This year we should be able to publish it. Next. So basically, with genomic tools, I would say, we've used different genetic resources, created genomic resources from there, whether it is the reference genome, the whole genome sequencing, or functional omics, or even pangenomic platforms, we have been able to create that, and tried to bridge the gap between the genotype and the phenotype, so that the plant breeder can use these for translational approaches and so forth. That means you can make informed decisions. These are fact-based decisions, not just like that. Yes, you know, you have proof for that and you can take those decisions. Next slide. So it has been a long meandering career, I would say. It's never been a straight line, never is, but we have never stopped. We have moved here and there like the water. When it faces a rock, we say, okay, we'll make a new way, move there. So I think in the end, it is all about going in the right direction rather than making something. Even Google Maps are wrong these days, everyone says, but to have a compass is more important, you know, you're heading in the right direction. Thank you so much. I may have left many questions, created more queries, and I would be very happy to address some of them if it is possible. कोई question नहीं?
Any questions?
Okay. No, I'm always under scrutiny for many things, and I get to learn from a mixed audience also.
Those who wish to discuss?
Yeah, you're most welcome, most welcome. I would be happy to have your inputs.
Thank you, ma'am, for
Thank you. I hope I've not overstepped.
Thank you. Yes, ma'am. Thank you, ma'am, for such an insightful and thought-provoking lecture. We are truly grateful for your valuable guidance and inspiration. And once again, I request Pro-Chancellor Arun Pandey, sir, Vice-Chancellor A.S. Yadav, sir, Pro-Vice-Chancellor Rohil, sir, Akhilesh, sir, please take their respective seats on the dais. We now move to next step of our program, that is citation and the felicitation of our chief guest. Traditionally, the citation is read by the Secretary of the Society. However, as the Secretary is unable to be with us today, I respectfully invite Pro-Chancellor Mansarovar Global University and former President of the Indian Botanical Society, Professor Arun Kumar Pandey, sir, to kindly read out the citation.
Now we are coming to the end of the program, but before that, I would read the citation of the Foundation Day Lecture 2025 of the Indian Botanical Society. It is accorded to Professor Paramjit Khurana, FNA, FASC, FNASC, FNAAS, FTWAS, INSA Senior Scientist and Vice President, National Academy of Sciences, India, formerly J.C. Bose National Fellow, Senior Professor and Head Department of Plant Molecular Biology, University of Delhi, South Campus, New Delhi. During the felicitation function and the lecture delivered at the Mansarovar Global University on 10th February 2026. An elected fellow of the major science academies, Professor Paramjit Khurana, has made pioneering contributions in the area of wheat and seri biotechnology and comparative genomics. In addition to an active participation in sequencing of chromosome 11 of rice and chromosome 5 of tomato, as a part of international initiative, her group has sequenced the complete chloroplast and nuclear genome of Indian mulberry and was involved in sequencing of chromosome 2A and subsequently complete genome of wheat as well. During past 2 decades, genetic transformation of Indian wheat has been accomplished by her group for resistance against the cereal cyst nematode and for drought tolerance. Mulberry transgenics capable of withstanding salinity, drought and high radiation leading to stress tolerance in crop plants were developed and transferred to CSIR TI Mysore and US GKVK Bangalore for sustainable agriculture under changing climatic conditions. Her initial research pursuits were focused towards understanding the molecular basis of somatic embryogenesis and later on heat stress tolerance in wheat. She has authored over 200 publications and presented equally in conferences and workshops. A recipient of J.C. Bose National Fellowship and Birbal Sahni Medal of the Indian Botanical Society, she has been awarded the Archana Sharma Memorial Award by ISCA and NASI, A.K. Sharma Oration Medal of the Archana Sharma Foundation of Calcutta, Sri Ranjan Memorial Lecture Award of NASI, S.K. Sinha Memorial Lecture Award by the Indian Society of Plant Physiology, Dr. S. Rajarama Outstanding Women Scientist Award by the Society of Advancement of Wheat and Barley Research, O.P. Bhasin Award in Biotechnology, INSA Distinguished Lecture 2. It is signed by the Secretary of the Indian Botanical Society, Professor Mrs. Sheshu Lavania, and President Professor S.K. Barik, who is presently active Vice Chancellor of Northeastern Hill University, Shillong. Madam, we would like to present the citation to you.
Thank you, sir, and I respectfully invite all the dignitaries to kindly facilitate Professor Paramjit Khurana ma'am as a mark of our deep respect, sincere gratitude and appreciation for her remarkable contribution to the field of botanical science. Thank you so much, sir, ma'am. And to formally conclude today's program, I respectfully invite our Honorable Vice Chancellor, Dr. A.S. Yadav, sir, to kindly propose the vote of thanks.
Thank you. On behalf of the Chancellor of Mansarovar Global University, Mrs. Manjula Tiwari, the CD and Pro-Chancellor, Engineer Gaurav Tiwari, and on my own behalf, I express my deep sense of gratitude to Professor Paramjit Khurana for graciously accepting our invitation and delivering the Indian Botanical Society Foundation Day lecture. Her insightful deliberation on plant genomics for climate resistant crop has greatly enriched our understanding and inspired scholars, researchers, and students alike. I extend our sincere appreciation to the Indian Botanical Society for its continued commitment to providing botanical science and fostering academic excellence across the country. I am grateful to Professor Arun Pandey, Pro-Chancellor, Mansarovar Global University, for meticulously planning the Foundation Day lecture and getting it nicely executed. My heartfelt thanks goes to Professor Akhilesh Pandey sir, former chairman and former Vice Chancellor, Vikram University Ujjain, Colonel H.R. Rohil Sahab, Pro-Chancellor of the University, Dr. Pushpendra Tiwari, Registrar, Professor H.K. Goswami, former Head, Department of Genetics, Bhopal University, Bhopal, Dr. M.K. Tripathi sir, Principal Scientist, CIE, Professor Rahul Srivastava, Manit, Bhopal, Principals and distinguished faculty members, researchers, and students whose enthusiastic participation made this lecture a meaningful academic gathering. Your presence reflects the vibrant intellectual spirit of our university. I also wish to acknowledge the dedicated of organizing committee and supporting staff whose meticulously planning and hard work ensured the smooth conduct of this event. Finally, we are grateful to the university management for their constant encouragement in organizing such scholarly interaction that contribute to knowledge creation and dissemination. Thank you all for being part of this memorable occasion. We look forward to many more such enriching academic engagements in future. Thank you. Jai Hind, Jai Bharat. इस प्रोग्राम में जो हमारे एंकर थी मैडम मीनाक्षी, उनको भी धन्यवाद। उन्होंने बहुत बढ़िया ये प्रोग्राम एंकर किया, बढ़िया तरीके से प्रस्तुत किया। सभी को धन्यवाद। थैंक यू।
Thank you so much, sir, for this kind gesture. And now I request all the faculty members and all the members present in this hall to please stand up for the national anthem.
शुभ नामे जागे, तब शुभ आशीष मांगे, गाहे तब जय गाथा। जन गण मंगल दायक जय हे, भारत भाग्य विधाता। जय हे, जय हे, जय हे, जय जय।
With this, we came to the end of today's Indian Botanical Society Foundation Day lecture program. Thank you all for your gracious presence and cooperation in making this program a grand success. I now request Registrar Sir, Principal, Deans, heads, and all other faculty members, please come forward for our group photographs.